Hello, this is Tommy. Welcome back to Genomics. Today we will continue to replicate figure 1 of a genomics paper. So make sure you watch all the previous videos in this playlist so you understand what's the project about. So all the links will be in the description of this video. The markdown files, you can follow all the analysis. Okay, let's take a look at figure 1D, E, and F. So figure 1E is a wet lab experiment, so we're not going to reproduce this one. But for figure D and F, let's see what kind of figure type are they, and then what data are needed to plot them. Okay, for figure 1D, and you see this is just a scatter plot, right? Then you have x-axis and the y-axis. And then you see those dots, essentially each dot is one peak. And the peak is the overlapping peaks among actually these three factors, YAP, TADS, and the TAD. So the paper of the authors did ChIP-seq for those three transcription factors. And essentially you need to first get the overlap of those three peak sets, and then you need to count. So essentially, this is the TAD4 signal on the x-axis and the YAP1 on the y-axis. You need to count how many reads in each of the overlapping peaks, and then you can make a scatter plot. So this one just, because you have three factors here, it's just a pairwise scatter plot. And figure F here, it shows you, first of all, it's a stacked bar plot, so it's just a bar plot. But the y-axis here is to show you the percentage of those peaks, actually YAP, TADS, TAD, and also the overlapping among those three sets. So what's the percentage of the peaks that is, for example, smaller than 1 kb, the distance to TSS, or 1 to 10 kb, or even greater than 100 kb. So you can use, for example, packages like ChIP-seeker to do this easily, but I will show you how to do that from scratch. Okay, let's go ahead. Okay, so let's load the library first. Genomic ranges to deal with intervals, R-track layer to read peak files or export peak files. Here it's just to construct the relative path so you can read the files. Dplyr and ggplot2. Okay, so we already actually called peaks using max3, and it's in this folder. So I will just use the import function from R-track layer to read the peak, TADS, YAP, and TAD4. Okay. And once we have that, we can, for example, find the overlaps. So now we will find the YAP1 overlap with TADS. So you use the subset by overlap function from Genomics ranges package, and then it gives you actually you need to input two genomic ranges object. So if you do that, okay, it will have some warnings, but it's fine. There's some unconventional chromosomes here. And this will be the overlapping sets, YAP overlap with TADS. Okay, so now you actually get, for example, 7,164. But to begin with, the TADS peaks, you have more than 10,000. Right. Okay, so once you do that, you can further overlap with the TAD4 peaks, right? So now use this, it's here, and subset by overlaps. Then now you subset this with the overlapping peaks for TAD4. So now you do it again. Now if you look at this, now it's getting fewer and fewer peaks. Now you get only roughly 6,000. Okay, now you can actually export this genomic range files into a bed file using the export function from R-track layer. So it will be actually exported to this file. Okay, so if you go to the terminal here, and if you say the bed files, okay, see where is it? In the FASTQ. Okay, so in the So now I'm actually in the main directory, the home directory here of the project. So go to data and go to FastQ folder, and then look at the bed. So actually I already have this file generated. Okay, the comment. So we can actually verify how many peaks are there, right? What count, space, yep, tat, comment. It's 5965. It's the same as here, right? And if you look at the file, it's a bed file, but it's 1, 2, 3, 4, 5, 6. It's a 6-column bed file format. But essentially, for a bed file, minimally you need 3 columns, chromosome start and end. Okay, with that file, now you can actually count how many reads for each of this peak from those 3 transcription factor actually band files. Okay, so we can use actually multi-coverage, and this from bed tools. Okay, so you can use this commands, bed tools, multi-coverage. Now you specify the band files. So this is the 3 band files. Okay, the YAP1, TAT, and TAT4, and then specify the intervals. It's the common intervals that we just did the overlapping, and then it will give you the counts. Okay, so this will actually take a couple of minutes. So I'm not going to run it, so I already ran it before. So if you look at this file, it's already generated here, but if you look at this file, it will give you essentially the same peaks like here, but now you have 3 additional columns. Those are the number of reads in each of those intervals from these 3 band files. Okay, from these 3 band files, YAP, TAT, and TAT4. Okay. Okay, but with that, we also, those are the raw counts, but each experiment or each transcription factor, they have different sequencing depths, meaning they have different number of total reads in each experiment. You want to normalize it to, for example, counts per million, so you can kind of compare between different experiments. So to do that, we need to get the total number of reads for each transcription factor chipseq. And to do that, you can use SAM tools flex stat. For example, if you use this. Okay, I think I need to activate the conda environment. I forget which environment that I have. So it should be this one. So conda activate. Okay, so now I can just use the up arrow sign to recycle the previous commands. So the same tools flex stat will actually show you the total number of reads and some other statistics. For example, it will tell you, okay, this total number of reads, okay, this number of read mapped, this total number. So we need to use this number, for example, for total number of reads that is mapped. So essentially, for example, each column here, the first one is YAP1. So the first actually is YAP1. So okay, the first column here is YAP1 reads for those intervals. And then you need to divide this column to this number times 1,000,000. So you get the counts per million, the normalized counts. Okay. So let's do that. To get all the numbers for those 3 band files using the same commands. And then this is the number you need. And then also you do the same thing, and this is the number you need, the total number of reads that is mapped. Now we can read that back into R. So this is the total number of counts. So if you look at the counts here, so those are 3 counts. So now you give some name for those columns, chromosome start end, the name, the peak name, the score, whatever, then the value here, and then you have the YAP1, TAT, and TAT4. Take a look again. So you have the column names. Now we can normalize to counts per million. So remember those 3 numbers we got from SAM tools flex stat. Then we can just mutate this column divided by the total number of reads mapped times 1,000,000. The same thing for those two columns. Now we have the normalized counts per million. So with this data frame in hand, you're ready to plot that scatter plot, right? So you now, for this two, then you just this column versus this column, right? So you can do TAT4 versus YAP1, for example. You can do that. And just give you the scatter plot. And you notice that there's one outlier here. It's like really big signal for TAT4 and the YAP1. Right, so if it's like roughly 60 here, right? So you can actually find that peak. You can do that, and you see this is the peak region. And like whenever you see something that is abnormal, always be suspicious. So in this case, try to look at IGV whether it's something strange there. So I actually looked at IGV, and then it looks like it's real, and it's not in one of the blacklist regions like from ENCODE. So if you look at here, I think I have a screenshot here. Yeah, I look at from IGV. This is the big week file. This is the peak file, and then I look at the specific peak here. It looks like it's real, and it's not in the backlisted region either. So. Okay. So what we can do, so we can actually either remove that outlier, or you can use the log2 scale for the x-axis and y-axis. So to do that, so you can just use ggplot to add these two lines, like scale x continuous and scale y continuous, and use log2, okay, and add some labels here. So if you do that, and you see this is the scatter plot we need. So what we still need is that linear regression line and then the R square, right? So we will use this ggPMIS package to add R square. You can probably also use ggpopr to do the same thing. But in this case, I will just use this function here from this package. Okay, so we'll add the R square. So if we do this, I need to load the library first. And then do that. Okay, now you get the R square of 0.66. Okay, so the correlation coefficient is which is the R, which ranges from -1 to 1, and this is just the R square. Okay, so if we calculate the correlation, actually it's 0.8, but if we square it, it's 0.65, so it's the same as 0.66 that is calculated by the package itself. So always verify whether that package give you the right answer. Okay. Now we can easily actually create the other scatter plot, right? Just change the x and y axis, x-axis and the y-axis. So because it's just a pairwise comparison. The same actually commands. Then we get the other scatter plot. So we reproduce figure 1D. Okay. So, okay, now let's reproduce figure 1F, which is that stack bar plot. Okay. So from the figure legend is that is this absolute distance of the YAP1 peak, TAT4 peak, or the overlapping of those three sets to the nearest TSS. So we just need to actually get the distance of those peaks to the nearest TSS, and then get a table, and then we can make that stacked bar plot. Okay, so let's do it from scratch. So now you need some of the annotation packages, like from transcript database, TXDB, like for example, load that, genomic ranges, genomic features. So now we can get all the transcripts actually using this transcripts function, and this is the database. If we do that, and this is all the transcripts actually for in HG38. So this transcript name, right? And what we can do, we also need to get the transcription start site. So the transcription start site will be the first space here, right? The start here if it's on the positive strand, and it will be this one, actually, for example, if it's on the negative strand, will be this, will be the TSS. You can do that like manually, but you can also use the promoters function and then use upstream equals 0, downstream is 1 to get the transcription start sites. Okay. So let's actually verify it. For example, the first one is 11869, so it's 11869. And for this one, it should be 260095. So it'll be 260095. Okay, so it's correct. So always do some sanity check when you're doing analysis. Now we can calculate the distance to the TSS, for example, for the YAP1 peaks. So this is a genomic ranges object. And this is also a genomic ranges object with length of 1 because it's 1 base TSS. So you can use distance to nearest this function from genomic ranges or genomic features package. And you can calculate the distance. And if you look at that, this is a hits object, okay? And this is the index of your query, like YAP1 peak. And this is the index of your transcription start site. We can actually access this distance column here, use the mcost function. So it gives you the distance as a data frame. You can, let's say, use this and then get this column, and then you get a vector of the distances, okay? Then we can do the same thing for all of them, right? YAP, TAT. Let's do that. Now we have all the distances, right? And we can do, now we can put them in the same actually data frame here. So convert the data frame, for example, like this. If you do this, will be the data frame, right? And then this will be data frame for TADs. So, and then let's bind the rows. So actually, it will give you a long format. So if you look at this, and now if you look at the original figure, it has this different categories: smaller than 1 KB, 1 to 10, 10 to 100, greater than 100. Right? So if we go here, so we need to actually make that. So we can actually add another column here, it says category, and mutate. Use the case1 function. So if that distance is smaller than 1,000, it's smaller than 1 KB. If it's 1,000 to 10,000, is 1 to 10. And then the 10 to 100 KB, and then greater than 100 KB, right? So if we do that, so we can actually add another column, so indicating the category of the distance. So you can see how I actually build those steps, like using the pipe step by step. And once you have that, now you can group by this factor and the category, and just count how many, right, in each category. So if you do that, then you get this data frame. So for YAP1, you have this many in this category and this many in this category, and then the same for TAT4 and YAP. Okay. And also you need to, because the original figure is a percentage, you also need the total number of peaks in each for all the category, right? So you can just use this and they just count. So the total for each transcription factors. So you get the total counts, save it into a new data frame, and now you need to merge those two by join. Now you have this. This is the number in each category, but then this is the total for the same transcription factor, right? Now you can calculate the percentage, mutate a new column, n divided by total times 100, and now you can plot using ggplot2, x equals factor, y equals to the percentage we just calculated, fill by the color category. So then the color will be based on the factors here. So, and I use the scales package, so you can format that as a percentage. If you do that, and now you have the percent. So the y-axis label will change to percentage because if I use this. Okay, so this is essentially we reproduce the same actually figure for figure 1F, but we can change the color to the similar colors in the paper, right? So I just use another function here, scale field menu, and specify the colors manually. So by the way, I use a tool, essentially I click on the figure and then it will tells me what's the hex code for that color. So it's an online web tool you can use. Okay, so if I do that, now this is exactly the same color as in the original figure. Okay, so the original figure actually shows a big proportion of peaks greater than 100%, 100 kb. So for me, if it's greater than 100 kb, it's like not that a lot, right? But if you look at the original figure, greater than 100 kb, like it's like 40%, like almost 40%. That's actually a little bit suspicious to me, because usually, unless those peaks they're all in the intergenic regions, you should not have so many peaks that are greater than 100 kb, right? So actually for me, this is, say, my 10 to 100 kb. There's actually a very small portion here, actually greater than 100 kb. Okay, I think my results makes a little bit more sense. Okay, that's it for today, and make sure you subscribe to this channel, and I will see you next time. And I will finish recording all the videos, reproducing all the panels of that figure 1. Okay, see you next time.